Jo ja l'estic veient per pantalla, però saludem a Coleen Murphy, professora de Genomics and Molecular Biology at Princeton University. She's connected from Georgia. Good morning for you, I imagine. Good afternoon is here in Barcelona. How are you?
Hi, how are you? I'm good
Hello.
today.
What time is it in Georgia now?
It's 9:30 in the morning.
So you start the week?
Yes.
We are very proud that you start the week with us. We are lucky because we've been talking a lot during the morning about longevity, and it's for us a privilege to have you with us this afternoon in Barcelona. So we are going to talk, and we are going to hear your keynote talking about how we age, the science of longevity. Podrem fer-li preguntes, té una speech de 30 minuts, so we can have some questions after the speech from Coleen. So thank you. You can start, and then we can have some questions for you. Go on. Thank you.
Thank you very much for inviting me here today. I'm going to tell you a little bit about the work that I and others in the field have done to better understand longevity. So, of course, all of us want to live longer, but really we want to live better. So you may know about some very famous examples. For example, Jeanne Calment, she's thought to be the oldest living human. But there are other people who have lived almost as long as Jeanne. So Sarah Naus and Kane Tanaka, there are examples of other people who have lived well over 100 years. But of course, we all know that we don't want to live just longer. We want to live better, right? So we want to have a better quality of life, not just quantity. And of course, when we think of aging, a lot of us think about the downsides. So we want to understand what are the mechanisms by which aging is regulated, and can we do anything to modify health span? And of course, when I talk to young people, they always ask me if it's ethical to study aging, and I think that we can answer that question very quickly. So, for example, if we think about all the people who are getting older, that's actually a good thing. It means we are doing a better job of keeping people living longer. But of course, when people are older, they actually get a lot of other associated diseases. So a lot of chronic and degenerative diseases. And so the idea that we have in the field is that if we can slow down aging, maybe we could slow down.
¿De verdad? ¿Qué va? Gracias.
Is everything okay?
Gracias.
That we hope that we can slow down the onset of all these diseases. All right. So one of the ideas in the longevity field is that we don't want to just live a long time. And so when we think about this, we think about morbidity. When do we start to get sick? And we don't want to just live longer without changing that. What we want to do is slow down and offset the time of morbidity so that we can compress that time, so that people are living a longer, healthier life, even if we don't change when they actually die. So in the field, we had a lot of different ways to study aging. And so many of you are familiar with centenarians. So these are people who live over 100 years old. And we can study the genes that are associated with that aging and long life in genome-wide association studies. Of course, those are not experiments. Those are just looking at what data exists in people. We can also look at extremely long-lived animals. So this is the bowhead whale. It lives to be over 200 years. But of course, we can't do any experiments in that. The other end of the spectrum, we have things like cells. So if we do things in culture, we can study a lot about the cellular mechanisms, but it doesn't help us understand better how long we can live. So I'm going to tell you about work that's being done in model systems. So that means that there's an animal that we can do experiments on. We can understand everything about the genetics and the cell biology that's going on. Okay, so there are a lot of different ways to study aging. We can study bacteria, which are super short-lived, but of course, those are not very much like us. At the other end of the spectrum, we have things like, as I mentioned, the bowhead whale or the Greenland shark, but we can't do experiments on those. And so what people in the aging field study are usually these short-lived organisms, such as C. elegans, which I'll tell you a lot about, worms, fruit flies, killifish, which is a new model system. So these animals all live days, weeks, or months. And then, of course, we often do other studies in mice to verify things, and then all the way up through things like monkeys, like primates. And so I'm going to tell you a lot about C. elegans because we've learned a ton about the aging mechanisms in C. elegans and use that to learn more about how we age. And the reason that we are so excited about this is because most of the proteins that these little worms have are the same as in us. And in fact, you'll know more about this if you think about the Nobel Prize in 2024. So we just won that again for another discovery in C. elegans. Okay. So when we think about aging, instead of just studying how long an animal lives, we can ask the question, are there facts that we can learn from animals that live a long time? Okay. So for example, we want to understand, why does one organism look younger and act younger than another organism of the same age? And so an example we showed in C. elegans is that these animals, they live for only 2 to 3 weeks. Okay. So in that short time, we can see them age. And so if you see here, we can see that C. elegans, just within 2 weeks, they start to look fat and slow, and they stop moving around so well, just like us. And so we can actually observe those changes. But the real thing that changed the whole aging field is this. Okay, so I'm going to show you a movie, I hope. Hope that's working. Okay, so this is C. elegans crawling around. They're really tiny. They're only 1 millimeter long. And this is the wild type. This is the normal type, okay? So we found a mutant that I'm going to tell you a bit more about called DAF2. All right, this mutant looks the same as it does when they're young. Looks just like the normal, the wild type worms, okay? But these worms only live 2 to 3 weeks, okay? So wild type worms, the normal worms, they are really sickly and they're almost dead by 13 days. All right, so you can see this, and then I'm going to show you a few more pictures of worms. So this, what I'm telling you is, anyone in this audience, if I took you to my lab right now and told you to look through the microscope, you'd be able to tell me that these worms look old, right? They look old and sick and dead. In fact, this one might be still alive. All right, but this mutant that we're going to talk about, called DAF2, looks like this on the same day of adulthood. Okay, so this is a worm with only a single mutation in its entire genome. And just that single mutation helped this animal live twice as long. Okay. And so if we look at just a survival curve, we can see that those worms, not only do they look young and healthy, but they live twice as long. And this was a discovery in 1993 that changed the whole field, because it turns out that that mutation is in the insulin pathway. And of course, all of us have heard of insulin and its relationship to diabetes and generally in health. And then when we looked at those humans who live to be over 100 years old, they have mutations in this same pathway. Okay, so now we know that those changes in that little worm actually have something to do with how long we can live. And so if you're a geneticist, you can see that a mutation in DAF2, this insulin receptor, it normally stops longevity, and it acts through a transcription factor, a gene called DAF16. Okay, so this is the genetics that we know. And DAF16 turned out to be FOXO, a transcription factor, which is also conserved in humans. And we know a lot about how that functions. Okay, so this transcription factor binds to DNA, and what it does is turns on gene transcription. And the genes that it regulates are in 2 categories. So it turns up genes that are very good about stress protection, antimicrobials, protein folding, things that help a cell live long. And it blocks genes involved in growth and development. All right. So it allows the cell to always choose between stress resistance and growth and development. And that's what our cells are doing all the time as well. Okay, so I'm going to switch gears a little bit, because most of you have heard of dietary restriction or caloric restriction, right? Because that's what you're always seeing advertised, intermittent fasting, things like that. So what does that mean? So dietary restriction is a condition under which an animal takes in less than 30% of its normal calories. So this is very little, actually. It's been really well studied. In every organism that has ever been studied, dietary restriction slows down, it extends lifespan and slows down aging. And in some systems, intermittent fasting, which you're probably familiar with as well, may be very similar. And what this audience may be interested in is there's many drugs that are being developed to mimic dietary restriction. And this would be a good thing, because it means we wouldn't have to actually dietary restrict ourselves. Because just as you see, These healthy-looking monkeys are actually not very happy. So, and I don't think we would be happy either if we had to dietary restrict. But it's a complicated pathway. There's lots of different ways that you can get extended longevity through many different ways of doing dietary restriction. So this is actually a good thing. And it means that we have many druggable targets to try to get to this extended longevity state. And in fact, if we look here, there's a lot of different, so the blue boxes, these are all drugs that are being used to try to inhibit different parts of this pathway. So it's actually a very exciting and active area of research. And we're trying to understand, are there ways that we could actually give people drugs that would make them healthier to mimic dietary restriction? Okay, the other thing you may have heard a lot about has to do with cell senescence. So cells can only divide a certain number of times, and that's called the Hayflick limit. And what that means is the cells can divide and divide and divide until they get too sick, and that's often because of these telomeres, okay? So it's usually 40 to 60 times. That's the limitation. It was discovered by Lenny Hayflick in 1961. And the reason it's important is because people had this idea that cells could divide forever. But now we know that's not true. And not only can they not divide, but when they get to the stage of becoming senescent, they become very ill, sick. And it's probably, we know that's due to telomere length. But what they do is they make other cells sick as well. So getting rid of these senescent cells might be important. So just to explain that a little better, what we know is that there's a phenotype called the senescence-associated secretion phenotype. And what that means is these senescent cells, not only are they dying, but they're putting out other things that make other cells sick, okay? And that can cause chronic inflammation, which we all know is a part of aging. And so the idea in the field is that maybe we could develop drugs that would target these senescent cells and get rid of them before they become toxic to other cells, okay? And so that's shown here. We have all these inhibitors that we can use to target these senescent cells. So that's another area of active drug development. Of course, there's a risk. There's some idea that if we turn off this pathway, there may be a risk of cancer. So we always have to think about the downside of some of these drugs. Okay. Another area of research in the field is on biomarkers. And the reason we want to study biomarkers is because we want to understand, can we tell if someone is getting aging, or are they aging more slowly? And importantly, if we give them a drug to slow aging, are we actually slowing aging? Okay. And this is just to give you an example of how we can tell someone's aging, right? So we all know that when we see people, we can tell approximately how old they are. But of course, someone who's led a really hard life, like Sharbagula, the Afghan refugee, she's had a really hard life. And so we can tell that she's aging more quickly. And so to find biomarkers, what we'd really like to focus on are not just chronological age markers, but biological. Can we understand how a cell is aging? And a really good biomarker would be predictive. It would tell us how much longer someone will live. All right. And then we could use those biomarkers to assess whether our drugs, our interventions are actually slowing aging. All right. And so there's a lot of different things being tested for that, blood proteins, hormones, et cetera. But as I just showed you, even faces might be informative. And so let me just share with you one interesting study. There's work by Jackie Jingyang Hong, where she showed that if she developed a 3D facial imaging method, she could actually measure the rate of aging in humans. And so that's been really exciting. She's been able to show that. And it turns out, let me just go back one, that if just by measuring these different marks on a 3D face, she could actually see whether there were interventions that might slow. And it actually correlates really well with blood biomarkers. And so that's an active area of research as well. But the thing that you may have heard most about are blood methylation clocks. I don't know if anyone in your audience has done this, but you can send your blood off to have it tested, okay? And the reason this works to some degree is because there's work being done on blood cells and other tissues where we look at the DNA and see these methylation marks on the DNA, all right? So from this famous paper in 2013, Steve Horvath looked at blood methylation marks. And what he found was that there's a small number that actually then change with age. And just by using that, he could then predict chronological age. Okay. All right, so there are a lot of these aging clocks, and you may get advertisements for these sometimes. What's to know about this is this last part. The accuracy and precision of these clocks is not yet perfect. And so I think that these will get better with time, but many are being sold commercially and they may not work as well as we had hoped. Okay. In humans, you can do another test, which is really what gerontologists do. It's called the short physical performance battery. And that is involved like basically doing these chair stands, balance testing, and a fast walk. And so what we were interested in was how well can you use one of these to predict future longevity? All right, so this is used by gerontologists, but we want to know whether you can do this in a model system. All right. So what we did with C. elegans was ask the question, what happens if you just ask how fast can these worms swim? So we did an experiment where we tested the worms every day. We video recorded them for 30 seconds, and then we did data analysis. And what we discovered was that if you track each one of these worms over time, their maximum velocity declines with age, which makes sense, right? So if I were to tell everybody to get up and run as fast as you could, then we might have some idea of how healthy people are. And in fact, we can see that worms that moved very quickly in middle age actually live long, and the ones that move slowly live short. And so the real question for us is, can we extrapolate that to humans? Okay, so what would be the equivalent to test in middle-aged humans? And we don't know that yet, but that would be a good question to ask. Okay, so I want to just sort of quickly go through some other things that we've learned in the field. We have some really big ideas that were kind of surprising at the time they were found. One big idea is that your reproductive system or your germline can inform how long you're going to live in the future. Okay. Another idea is that what you sense may also regulate how long you live. We've already talked about these clocks to measure aging. And at the end, I'm going to mention some of the things that you may have heard of, such as blood factors and the microbiome. All right. So, for example, we discovered in C. elegans that if you knock out the two cells that are going to become the animal's future reproductive system, that they then live long. So that's this red here. Okay. So that's not surprising because we think of that as being like a sterile animal. But what I want you to notice is blue means we've knocked out the entire reproductive system. And this tells us there's two opposing signals. This is not because the animals are sterile, because the blue line, they're also sterile, but it's because the animal anticipates what future needs it will have to reproduce and adjusts longevity accordingly. And so that's something we're interested in as well, is what are those signals? And so let me just tell you, if you add up all these pathways together, you can get animals that are super long-lived. So here's an example of these worms, which should only live for 2 weeks or 20 days or so. These are 114 days old. And so they're still living very healthy. Oh, sorry, 144, right? So this tells us we can add these pathways together. So the more we learn about these pathways, the more we might be able to adjust longevity and health. Okay, another important finding in the longevity field was that what we smell or sense can regulate how long we live, okay? And so we already knew this in C. elegans. I'm going to go through this very quickly. If we just knock out the neurons that affect how they smell, we can see that they live really, really long, okay? So that's just worms. What we figured out in the field is that this is true in C. elegans, but it's also true in Drosophila, so fruit flies, and the same systems work in mice. Okay, so it's not just some weird worm thing, and almost nothing I'm telling you is a weird worm thing. Everything is something that we've been able to find in other higher organisms as well. But one of my favorite studies is from a group where they were looking at Drosophila, and this is a crazy story. What they looked at was what happens to flies that can see other dead flies, okay? So they had flies, a Drosophila, that could see through a plexiglass window some other dead flies, and that stressed them out and it made them live short, okay? So you can think of this as an alarm signal. When animals see other dead animals, they want to get away from them. And this was completely based on their vision. Okay, so now I'm going to just, in the last part of my talk about some of the interesting approaches people have used to try to slow aging. So some of you may be aware of this idea of our blood containing signals that can control longevity. All right. And so this was developed out of an idea called heterochronic parabiosis. So this happens when you have two animals of different ages. So that's the heterochronic, and they're stitched together so that they start sharing a circulatory system. So there's good news and bad news. The good news is that the old animal receives signals from the young animal that can rejuvenate them. Okay. So that's something we can learn from. What are these rejuvenating signals? Of course, the bad news is that the old animal can give bad signals or factors, bad blood factors to the young animals. So it's bad. So what we want to know is what are the good blood factors in these youthful animals that can help us live longer. Okay. And so, so this is an active area of research. And I think actually we're going to find some really important factors and their blood proteins that may be able to be given to people to slow even cognitive aging. And if you listen to no other slides from the rest of my talk, this is something everybody can do. Exercise is almost universally super beneficial. And so if you just look at people who have exercised their whole lives, so this is just from an observational study of a runner's club, you can see that they are much slower to become disabled, just by doing regular intense exercises. So I definitely recommend that because we're understanding that better and better. And in fact, it's not only from the exercise itself, but there are blood factors in exercise. So for example, in this study here from Saul Vieda's lab, they exercised mice and they gave the blood from those mice to older animals. Okay. And so what they see is that if they take the blood from these exercise mice and give it to sedentary mice, it improved how their brains worked. Okay. So identifying these blood factors could eventually help those who cannot exercise. So there's actually a therapeutic aspect. But again, I just highly recommend exercise because there's all kinds of good things that it does that we don't even know about yet. All right. And the last little thing that you may have heard about is the microbiome. So that's the idea that there's bacteria in our gut. They help us with our digestion. And of course, they shift. Our microbiome changes with age and in response to diet and drugs, medications. And so what we would like to know is there something good about young microbiome? All right, so there was a really good study. This isn't done in killifish. So this is a short-lived fish, the African turquoise killifish. And what Dario Valenzano's lab did was they took middle-aged fish and they either wiped out their gut or they gave them, and then they gave them either age matched, so middle-aged microbiome, or youthful microbiome. That's the YMT. And what they found was that the fish with the youthful microbiome lived longer. Okay. So that's very exciting. It really does suggest that there's something in the microbiome from young animals that is good for longevity. And even more excitingly, John Cryan's lab in Ireland did a nice experiment where they did a similar experiment where they did this, what's called fecal microbiota transplantation. So they gave young fecal matter to aged mice. Okay, so this is the young to old microbiome transfer. And I'm not recommending this, but it gives us an idea that it might work. And what they found was that the old animals that received the young microbiome actually had younger cognitive phenotypes, right? So it helped their brain. So that's worth understanding how exactly this works. And there's some idea that we might be able to restore these through the metabolites from the young microbiome instead of course, giving the microbiome. Okay, so I told you about a lot of information from the aging field from a lot of different labs. What I want to tell you is that there's still ongoing work and we're really excited about that. Okay, so I personally am very interested in cognitive aging and neurodegenerative disease. I'm not so interested in longevity. And the reason for that is, of course, we don't want to live a long time without having a functioning brain. And I think it's really important for us in the field to understand, are those the same treatments or are they different? And there may be specific cures for brain aging that are separate from longevity, but there may be some that overlap. And so I'm involved in research that will better understand that. I also think it's really important for us to focus not just on really aged longevity, but also what are the things that are happening in midlife that affect our lives. For example, we're interested in reproductive longevity, reproductive aging, maternal mortality, which is a huge problem, especially in the United States, menopause, and the effects on aging as well. All right, in the last couple of years, it's become really important for us to understand better what is happening after COVID-19. Are we going to see an increase in neurodegenerative disorders in the coming years? And we already see there's an increase in inflammatory and autoimmune disorders. And I believe that those are going to overlap with a lot of the work that we do on aging and longevity. And so we need to better understand those as well. And finally, as scientists, we need to not stay in our little bubble. We're trying to understand like what the molecular mechanisms are, but it's really important for everyone to understand that there are ways that we can slow aging and cognitive decline that are different from what we might give to people for like medication for longevity. For example, in the United States, there's an epidemic of deaths of despair. This is overlapping with things like the opioid epidemic. And in the United States, of course, we have a big problem with gun violence. And so these are things that are social problems that are, we might think they're separate from molecular biology, but they are overlapping with our interests in trying to understand how to keep people healthy longer. What I'm excited about is there's some new evidence that things like the opioid epidemic might be slowed by even GLP-1 agonist treatments that we are currently, that's all the rage for obesity, but it's actually very helpful, it turns out, in other kinds of addictions as well. All right. So what I want you to take home from this talk is that even though you've heard a lot about dietary restriction in the press, it's not the only longevity mechanism. And that tells us that there's other ways that we could maybe help slow aging. The other thing that's going on is that old drugs are being repurposed. So things like metformin, which is an old diabetes drug, and rapamycin are examples of how we might be able to do that. What's interesting as an academic is that we need to move our work from our research labs into, to get out to patients. And the only way we can do that is by supporting longevity biotech, and those are thriving. And the reason they're thriving is because there's so many different research areas. So if we want to stop cells from dying, the senescent cells, there's lots of drugs for that, senolytics and senomorphs. I told you about blood plasma factors. Those are also being developed. I mentioned the GLP-1 agonists that we might be able to repurpose that are actually, I think that's going to be the true anti-aging drug that we're going to see soon because people are just living healthier lives. There may be factors, metabolites from the microbiome. I put a question mark because that's kind of overhyped, but I think there's something real there. There's regeneration factors that are looked into. And then I haven't had time to talk about like ISRIB, but that's another drug that looks promising. And of course, a lot of academic and biotech labs are doing unbiased screening for new compounds. Okay, so just to wrap up my talk, we're learning a lot about the regulation of longevity. I gave you a lot of information today, but there's a lot more that's left to be learned. And the reason that we can use these model systems is because these longevity mechanisms are actually conserved all the way from invertebrates to humans. Evolution happened, and we can use that to our advantage to try to understand how we can live better. And understanding these pathways is helping us to identify and develop drugs that could slow these age-related declines. Now, longevity drugs, maybe, I wanted to put a little idea out there. There's a lot of people interested in living long, and they're interested in longevity drugs, but there's other people who are suffering from diseases earlier in life that we could really help by treating them with these longevity drugs. And I think that's really exciting. And last but not least, exercise, I think, is really the thing that people can do now to help themselves more than any diet or anything else. And finally, we want to think about not just how to make a few billionaires live longer. We really want to help people live better lives and have some equity in that. And lastly, I think that we need to think harder about how to invest in our planet because there's no point in living longer if there's no place to live. And so with that, I would be happy to take questions. And I hope, and I apologize if I spoke too quickly. There's a lot of information, but I do like to share a lot of information from the whole field, not just my own lab's research. So thank you very much.
Thank you, Coleen. An applause, please. We are really impressed about all your explanation, and maybe next year you can come here also to share with us this day. And do you think our audience, me, will be able to double our life expectancy by now, or it's just a dream, a science fiction imagination by now?
Okay, well, doubling would be, I think, impossible right now. And we might be able to, what I think is more realistic and likely is that we will be able to identify drugs that can help us stay functional longer. And that, of course, will, like if we can make our brains work longer, but, that would actually be better than living twice as long, I think. Yeah. In fact, actually in mice, no one's made mice live twice as long. So doing that in humans would, I think, right now be impossible, even though we've done that in invertebrates already. Mm-hmm. So, but I think it is a really good chance that we could lessen age-related diseases with age, and I think that's super important.
Okay, you have some questions from the audience. Now is the moment. We have some minutes left. So you have anything that maybe you can ask to Coleen Murphy? We have her here listening. Also, we have some people with our microphones. Montse, are you with the microphone here? So you have a question from Montse Davan in BioCat. Maybe you know each other, I don't know. But she's going to ask a question for you.
Well, thank you very much. I'm almost positive that we don't know each other yet, but we might next year if you come face-to-face to this event. So thank you for your talk. I have a couple of questions. I don't know if I'm allowed to ask two. The first one is, so which mechanisms or hallmarks, I mean, you've been talking a lot about which are the developments now, but which could be like more a low-hanging fruit, let's say, for the clinical translation. And connected to that, if some of those are advanced therapeutics, is there like a consensus on how to regulate that to ethically decide who would have access to those therapeutics in the end? So those questions might be kind of related. So thank you for...
All right, those are great questions. Okay, so you might be aware of the studies right now to ask whether metformin is actually helpful as a longevity drug. In the United States, you can't study, the FDA doesn't allow approval of any age-related drugs because it doesn't consider aging a disease. And so there's a large study called the TAME trial, which is trying to ask the question, if you gave people metformin, would it actually slow their age-related diseases and make them live longer? And I think once that study is done, and there's similar ones like the PEARL trial for rapamycin, I think once that is done, we'll have better information about whether it's worth giving people those drugs. Now, drugs like metformin are super cheap, so I think your second question about who should get them actually has to do with expense, right? Because it's not a problem if the drugs are cheap. It's more of a problem if they're expensive drugs that need to, that we can't give to everyone. And so you're asking who should benefit most from that? That's a good question. I think the Europeans will probably deal with that better. The United States right now is a mess, and so I think that all those kinds of things are going to get really screwed up. I expect in the United States a bunch of billionaires will get access to drugs that regular people won't, and that's a terrible thing, and so hopefully that will be reversed. What I really think is a lot of these drugs, so I mentioned that some of the longevity drugs may be repurposed. So for example, so children who have had cancers treated, as childhood cancers, when they become middle-aged, they often suffer from premature aging phenotypes, lots of problems. And so I think those patients may be ones who could benefit most quickly from these kinds of drugs. For example, I understand that there's a trial going on with using these senolytics to treat some of these survivors of childhood cancers, because it seems like they have a lot of senescent cells. So that's where I think that you could argue those people should get the drugs, and that would be less of an ethical concern because they definitely are suffering from age-related diseases prematurely. Did that answer your question?
Yes, yes, perfect. Just I was going to ask you to add some things. If more than ethics, you find which barriers you have you think that you will have to get to reach the market quickly? Because I'm not going to tell you which is my age, but I need some to move fast in this case.
I'm not in biotech, but my understanding is there's several different approaches. Okay, so one approach that I don't love but is going on is that people are giving some longevity drugs as supplements, right? So those are not regulated properly. And so I think those are going to market. They're a market now. The other things that are more ethical, I think, are the drugs that are undergoing clinical trials, but not for aging, but for age-related diseases like osteoarthritis, a few other things. And so that's where drugs can get tested ethically. We know whether they're safe or not. And so there's several drugs that have gone through that process, but have failed at like phase 3. So eventually we'll get there, but that's what the biotech companies are doing right now. And then finally, I just wanted to mention the GLP-1 agonists. It's funny because people are very opinionated about those, but if you look at all the studies right now, it looks like that's basically doing what we would consider a longevity treatment, because a lot more people are healthy after they've been on them, because they lower their risk of cardiovascular diseases and all kinds of things. Some concern that I've heard from clinicians is that people lose muscle mass when they're on there. So all I can recommend is that if people are taking those GLP-1 agonists, that they also start to do some weight training, exercise, something like that, to make sure they combat this loss of muscle, because muscle itself is actually really good for us. It does a lot of things besides just make us strong. It actually puts things like factors into our blood that are healthy for us. So I think the GLP-1 agonists are actually already longevity drugs that will, at least in the United States where people are much more obese, that's actually going to raise the average lifespan.
Yeah, but as you said, exercise and diet, that means effort from our side. And taking drugs, it's easier, in that way.
Yeah, of course, there's expense, and a lot of these drugs are injections. And so I think you have to be really concerned about your health to take some of them. And there's some side effects as well. I think really what we're going to figure out, what I'm really focused on, of course, is the cognitive decline. And I think that those are the ones where we really want drugs to be able to help us sooner, because it's clear that Alzheimer's disease drugs, for example, right now are not working. And so we really need to find other treatments that will be useful. Yeah.
There's any other questions in the audience? There, upstairs? No? So, here, you have another question. Coleen?
Hello, Coleen. Thank you very much. Very nice presentation. My question is related to the conditions, or what needs to be true in the society, in a context, especially in the region here in Catalonia. What needs to be true for the authorities' regulation to speed up the processes and R&D in longevity? Because you mentioned that there is different regulation in the US. So my question is here, what needs to be done and what needs to be true to make that happen? Thank you.
Okay, so I think that very few countries consider aging a disease, and maybe that's okay because it's also very slow to do clinical trials on aging. And so I think that if people are interested in promoting longevity drugs, the way to go would be to identify age-related diseases that they think are good biomarkers for aging. There's a whole host of those, macular degeneration, osteoarthritis, cardiovascular disease. And so those kinds of diseases can be used as proxies for aging. And I think that if that's considered, then it could have both the effect of helping people with those problems as well as promoting the acceleration of longevity drug studies and acceptance.
And there's any other question? I think I saw somebody, no? Coleen, you talk about osteoarthrosis. Do you think that will be also possible even to reverse the consequences of aging?
I do think that it's interesting to know for every different age-related disease, once we understand the mechanism, then we can better answer your question. For example, for certain diseases where it's due to cumulative damage, it might take some time to just undo the damage, right? And some things may be irreversible. So it really depends on what kind of disease it is and what mechanism. But if it's about cell replacement, for example, you could imagine putting in stem cells and it actually would essentially reverse that problem. And so I'm not a big aging reversal person. I don't know about that a lot, but I do think there's specific mechanisms. I've been really impressed, for example, this crazy notion that people have of putting their own stem cells back into joints. I was very skeptical at first, but I've seen some results from that seem pretty amazing. So maybe for the types of aging mechanisms where it just requires that you get new stem cells to replace the tissue, you could imagine reversing that. It's harder to imagine for other pathways where you never have new cells. It's just the same old cells your whole life, like in your neurons or your heart. And so those may be not reversible. So I think we better need to understand all the mechanisms, the biological mechanisms, to then get an idea of what can be reversed and what cannot.
Thank you very much, Coleen. And do you think the investigations and all the trials you are doing in your research, are you doing any of the research with European scientists also, or it's only a states investment and trial?
That's a good question. I'm just about to write a grant with Europeans, and I think that actually American scientists are going to need help from Europeans very quickly.
Ah, wow.
So I think that's going to become a really active area of investigation because, politically it's a mess here. And so the administration is destroying all of our science. I don't know if you've kept track of the news, but the last 3 weeks in the United States have been a disaster for American science. And so we're hoping that something will help with that, and maybe more partnerships with Europeans will be the way to go.
So we wait for you. So that's a good proposal. We thought it was only our impression that scientists could be also affected by Trump new era. No, so you're not optimistic in that way.
No, not unless the courts stop him.
Thank you. If there's not any other question, just thank you very much, Coleen Murphy, for staying with us for a while. Have a nice week. It has been a pleasure for us to have you here with us talking about longevity. We're going to talk a lot more about that, and we are going to read your book. Some of the audience have already read the book because we've been talking about your book in the post. So thank you very much, and go on with your investigation. Bye.
Thank you.